Welcome to LabVantage's solution webinar on solving your laboratory analytics challenges. I want to thank you all for making the time to join us today. My name is Alan Marcus, and I'm the Chief Growth Officer here at LabVantage. Bioanalytics research in R&D labs often struggle due to lack of integration of analytics tools with their lab information systems. This makes the process inefficient and vulnerable to errors. LabVantage's integrated assay analytics solutions built on tcgmcube is automating and streamlining this for some of the largest biopharma and biotech organizations. Today, we will explore how AI and large language models can transform your research, speed up assay analysis, and minimize manual interventions. We'll understand how our integrated assay analysis library and advanced analytics can manage large volumes of documents and enhance reporting. And we'll learn how our solutions can drive higher quality and innovative outcomes in your R&D labs. Now, let me introduce our distinguished speakers. Dr. Krish Ghosh, President, TCG Digital, and Panchali Roychoudhury, Senior Director Consulting, TCG Digital. Dr. Ghosh has over 3 decades of experience as a distinguished business leader across diverse sectors, including pharmaceuticals, CROs, molecular laboratories, and data analytics companies. He's held senior management roles at MEBA, DeepThink Health, Semaphore, LabCorp, Covance, Pfizer, and BMS. At TCG Digital, he leads the Life Sciences division, transforming labs, clinical trials, manufacturing, and commercial processes with advanced analytic solutions. Ms. Roychoudhury brings over 22 years of expertise in analytics and data science consulting, project management, and solution development with a strong focus in life sciences. She has worked on solutions for pharma and biotech, as well as CROs, focusing on solutions for research, clinical trials, and manufacturing. At TCG Digital, she plays a pivotal role in the conceptualization and development of innovative AI-based solutions and products in the life sciences space. Now, thank you all again for joining us today, and please keep in mind you're all muted, but you can type in questions at any time in the Q&A section, and we will address those questions at the end of the webinar. You'll also receive the recording, the slides, and demo links post-webinar. So, without further ado, Dr. Ghosh, Ms. Roychoudhury, I turn it over to you.
Thank you, Alan, for the kind introductions. This would be an incredible webinar, so I'm glad that you have taken out the time to join. But to make this webinar really interesting, both Panchali and I are going to do a role play here, change our affiliation and job titles to make this webinar interesting. So here it is. Good morning, good afternoon, and good evening to all our participants and viewers. This is Dr. Krish Ghosh. I'm the Vice President of Drug Discovery at Cutting Edge Pharma. I have my colleague Panchali Roychoudhury with me. She's the head of all lab operations at the same company. Panchali, it seems you really like the lab analytics modules of the lab solutions. And this is driven by AI and want to purchase them for all our labs and replace the existing statistical and BI tools that are currently used in lab operations. But I'm not sure this is really going to add any incremental value. Also, we spent so much of efforts and money several years ago, almost 3 to 4 years ago, and it seems to be working fine. Am I missing anything here? Please help me understand the rationale for this decision.
Sure, Krish. So let me give a context. This whole intersection of AI and life sciences has brought in a new age of possibilities in transforming this whole medical research landscape. And this leads to huge opportunities for our R&D labs. Some of the key questions that I get from our scientists and our lab colleagues are all related to driving higher productivity and faster drug discovery. Currently, our lab systems are not integrated with any analysis software. We use third-party software, and this leads to a lot of manual effort in data handling. This not only increases analysis time, but also increases the risk of errors that we run. They ask me questions like, I put some of the questions here. So how do I get an integrated environment for analysis? How do I get a holistic view of the R&D data in the lab? How do I predict the workload using optimal scheduling? Or how do I balance workload across instruments, to balance this whole utilization of instruments? How do I benchmark to maximize ROI across the labs? How do I quickly search across different journals and scientific research papers and documents to get comprehensive insights? So AI has the capacity to process vast amounts of data and discern complex patterns and make predictions and recommendations based on these patterns detected. And this has set the stage up for the life sciences industry to witness revolutionary achievements. Hence, we need to move towards driving AI-based solutions for our labs.
Okay, I agree that we want to remain in the cutting edge. That's our company's name and mission, and be in the lab operation leadership role in this space. But why LabVantage Analytics? There's so many companies out there that promise the same functionalities as LabVantage Analytics. Isn't that the case? What is differentiating LabVantage Analytics from other software?
Let me first provide you with an overview of the LabVantage Analytics software, and then I'll talk about the differentiation that it has. So LabVantage Analytics comes as a module of LabVantage LIMS that extends the LIMS capabilities to cover end-to-end analytics. And it offers out-of-box BI and AI capabilities, as well as, and this is really important, it also offers a range of solutions to solve specific business problems in the lab, be it assay analysis or a Gen AI-based search, or driving different kinds of exploratory analysis on the data. There are so many different solutions. It is fully modular and can be implemented at different levels of adoption. So it can read data from the LIMS system and other data sources in the lab, including instrument data, and provide comprehensive insights on that data. Now, let me take you a little bit through the high-level architecture of this platform. So this is powered by a very powerful analytics platform. It's a robust end-to-end analytics platform called tcgmcube, which is powering for the whole LabVantage Analytics. So I'll walk you through this from right to left. On the rightmost side, you have the insights layer, which provides the dashboards, the interactive screens, the different reports, and this is the layer with which all our lab colleagues will interact with. This layer provides easy self-service capabilities, as well as a wide assortment of visualization options and drag and drop authoring and creation of new dashboards and widgets. So it's a very seamless user experience for our lab colleagues. Now, behind this robust analysis layer, that's where all the action is. That's where you have an assortment of over 1,000+ analytics and AI ML algorithms that support cutting-edge analysis. So things like deep learning, large language models, you name it, it has all of these different algorithms. The best part, it's powered by Spark, and Spark is a mainstay of the platform, and this technology enables it to crunch through large volumes of data using distributed processing to drive faster insights. Now, the solution does offer a big data store that supports different kinds of data. So in our labs, we have structured data, we have unstructured data, and we have large volumes of data. So what the data store does is it supports the creation of hot, cold data repositories. So data that is used frequently is what we call hot data. And this data is stored with in-memory analysis options for rapid search and query response, indexing on write for real-time analysis, distributed query processing for fast response, and extreme performance with very high scalability. Now, this layer can help manage master data and address data quality issues. You know, we have that a lot, and we have to deal with a lot of data quality issues. So this offers us inbuilt data profiling capabilities and will help us cleanse the data real time or in batch using a variety of transformation options. To fetch the data from different systems in the lab, we can leverage the connectors in the data ingest layer. So the data can be ingested in batch or real time, and the ingest can be triggered based on a time or an event trigger. The platform's modular and can work with the technology ecosystem which we already have in place in our labs. So let's summarize the building blocks that make up the platform. So these are the 4 building blocks that we have. So there's data integration, I call that ingest, then there's store, then there's analyze and visualize. And with these 4 fundamental building blocks, the platform has in its offering many different solutions which are built on top of the platform. Now, all the solutions may not need all the 4 building blocks, but if we have these 4 building blocks in place, any solutions can be churned out using these 4 fundamental building blocks. And of course, there's a very strong layer of security, audit, and so on. So this is this end-to-end analysis capability, which I just spoke about. This presents a huge differentiator with respect to what we have today in our labs. And it provides a clear advantage over some of the other platforms that I have been evaluating for our labs. To provide a seamless user experience, and you'll really like this. So our scientists are very used to using LIMS, and they want a very seamless user experience, and this exactly offers that option. So as you can see from the screenshot, the users can access this LabVantage analytics module right from the LIMS system. It offers a single sign-on with LIMS and can be directly accessed from a standard LIMS menu. The users and their access control can be defined in LIMS, and this information flows in seamlessly to the analytics module. So you don't have to define the users twice.
Okay, this is quite exciting. Okay, I wasn't expecting this. Can you talk about some of the differentiation, taking some specific examples or even just one example, if that's possible?
Yeah, sure. So let me walk you through the differentiation offered in more details by explaining how LabVantage Analytics helps with assay analysis. That seems to be a very popular solution. So I'll just walk you through this schematic that we have here. So the integrated assay analysis solution is built on that robust foundation of tcgmcube, which we just spoke about, which is that end-to-end AI platform that powers data analytics, offers transformative approach to assay analysis. So the solution has a data acquisition layer that provides seamless integration with our lab data sources, such as LIMS and ELNs. And can also read data directly from instrument files. The data analysis engine then analyzes the data, performing statistical and mathematical modeling as needed for sample analysis and for method validation. The solution provides access to multiple curve-fitting models and supports various user actions for analysis. The decision engine component then applies the relevant sample acceptance and plate acceptance and zone acceptance criteria to tag the samples and the plates as a pass or a fail. And this integrated solution facilitates a write back of the results to the lab transaction system for further action by the lab analysts. Now, there's an integrated reporting engine that generates the relevant reports by fetching the relevant outcome from the lab systems or from the solutions data store. The integrated solution has audit capabilities built in at every step, and the integration ensures zero manual data handling and manual data transfer from one system to the other. And this significantly speeds up the analysis process and reduces the risk of errors.
Now, this sounds interesting. However, as you know, we have so many labs, right, in our company, and we conduct so many different types of assays. How do we know that LabVantage Analytics can cater to all our needs?
So the platform actually offers a whole suite of assay analysis solution for the pharma industry. Let me show you. So the assay analysis solutions that you see here effectively caters to the different stages of the drug development, right from in vitro biology assays, in vitro ADME assays, PK studies, and so on. So it covers bioassays, ligand binding assays, immunoassays, ADA, anti-drug antibody assays, PCR assays, and many more. So these solutions support multiplex analysis, surrogate analysis, and many other different options that we need for our labs. So the assays will effective that you see here covers the need for therapeutic areas that we work in. So oncology, immunology, genomics, microbiology, and even platforms such as mRNA. So the method validation solution is also there. So it's not just sample analysis. So this solution includes various experiment options covering system suitability assessments, things like sensitivity, specificity, selectivity assessments, that intra-run and inter-run precision analysis, things like dilution linearity, dilution integrity, validation of negative controls, and so on. So the integrated reporting options generate standard M10 or other regulatory reports with zero manual interventions.
Now, these make sense to me, but can you walk me through a typical quantitative assay in a little more details so I can relate?
Sure. Let me do a deep dive on the PK assay analysis solution. So the PK assay analysis solution enables the users to perform the necessary quantitative and assay analysis in that integrated environment, reading data seamlessly from the LIMS and writing back the computed outcome back into the LIMS system for further analyst action. The solution provides a set of pre-packaged statistical algorithms for generating the standard curve using known concentration of the analyte being assayed. So they have linear regression options, non-linear options, so quite a few options available. And it offers options for multiplex analysis, offers options for surrogate analysis, and complete flexibility to our scientists, which will definitely facilitate easy adoption of the solution. This comprehensive solution is more consistent, efficient, and easier to manage, translating to increased efficiency for our lab scientists and significant cost reductions for our labs.
Okay, thank you for going through that. But definitely this makes sense on paper, but it would be great to see a demo or video of this product. Do you happen to have that?
Of course, here is a short demo.
Scientists in R&D labs lack an integrated environment for assay analysis, with little or no integration between LIMS and the analysis software. This makes the process inefficient and vulnerable to manual errors, leading to loss of productivity. Here is a short demo of our PK assay analysis solution that enables users to perform the analysis in an integrated environment. The solution fetches data on demand from LabVantage LIMS. You can choose the study method or the plate to be analyzed, and the system will fetch the data from LabVantage LIMS. You can choose from a library of linear and nonlinear regression options and also choose the desired residual weightage. You can also choose to perform the analysis at the replicate level or at the sample level. The solution offers various scenario creation options. You can specify the limits of quantification, anchor points, or go with default options. You also have the flexibility to mask various calibrators. Clicking on Run starts the analysis. The solution generates the standards curve along with the run acceptance parameters and the regression coefficient table. It also provides several tabular and graphical outputs. It applies the standards curve to generate the back-fit concentration for all samples on the plate, calibrators, QCs, and unknowns, and applies the relevant method specifications for sample and plate acceptance to tag samples, zones, and the plate as a pass or a fail. You will have the flexibility of masking different QC samples and specify the anomaly type for unknown samples, choosing from a drop-down of various options for sample and analytical anomalies. Clicking on Submit writes the results back into the LabVantage LIMS system using standard LabVantage LIMS APIs, thus completing the integrated bidirectional flow. The integrated solution has audit capabilities built in at every step and ensures a faster turnaround for analysis with a reduced risk of errors, leading to over 1 million expected cost savings for the lab. Try our integrated and comprehensive assay analysis solution today to get a significant boost to your lab's productivity.
Wow, I'm getting super excited now. Can you show me another one where we have been doing qualitative assay analysis? That'll be helpful.
Sure. Let me do a deep dive on the anti-drug antibody, the ADA assay analysis solution. Now, this solution, again, enables the users to perform the ADA assay analysis in an integrated environment, seamlessly reading data from LIMS and then writing back the computed outcome to the LIMS system for analyst action. But here there's a complete workflow because the ADA assay analysis has different tiers. So it covers the complete workflow needed for ADA assay analysis: screening, confirmatory, titer, and neutralization. And at each stage, it offers bidirectional integration with LIMS. It works with defined samples and plate acceptance criteria, along with the cut points from method validation, so that this solution can even help our scientists calculate the cut point with options for both parametric and non-parametric calculations. Now, before you ask for it, here is a short demo.
Thank you.
Scientists in R&D labs lack an integrated environment for assay analysis, with little or no integration between LIMS and the analysis software. This makes the process inefficient and vulnerable to manual errors, leading to loss of productivity. Here is a short demo of our anti-drug antibody assay analysis solution that enables users to perform the analysis in an integrated environment. The solution supports the complete workflow for ADA assay analysis: screening, confirmatory, titer, and neutralization. To start the analysis, the user can select the tier, the study, the method, and the plate to be analyzed and click on Fetch. The solution fetches the data on demand from the LabVantage LIMS system. Clicking on Calculate Results performs the necessary analysis using sample and plate acceptance criteria and the immunogenicity cut points from method validation. It applies the sample acceptance criteria to generate the sample status, and for every sample that is tapped as a pass, it applies the normalized cut points to tap samples as positive or negative. At any stage, you can view the analytical rules applied or configure the display precision for the several parameters. Clicking on Submit writes back the analysis outcome to the LabVantage LIMS system using standard LabVantage LIMS APIs, thus completing the integrated bidirectional flow. In a similar manner, you can also choose to do the analysis for the titer tier, calculating the titer cut points, and you can also choose to do the neutralization analysis and get the required outcome. The integrated solution has audit capabilities built in at every step. It ensures faster turnaround time for analysis and reduced risk of errors, leading to over 1 million in expected cost savings for the lab. Try our integrated and comprehensive analysis solution today to get a significant boost to your lab's productivity.
Now, no doubt, this is super cool. I think the scientists in your lab will really appreciate the new tools. But as you know, we use even more sophisticated analytics, such as flow cytometry solutions, and these are very difficult to build. Only a few companies can do that. Does LabVantage Analytics offer a similar type of solution?
Yes, let me talk about that a bit. So, there are many challenges for analyzing flow cytometry data in our labs. So the high dimensional panels that we have, the manual gating that we use, it introduces bias into the flow cytometry analysis, as it only identifies cell subpopulations deemed important by the analyst. So as the number of these possible subpopulations grow exponentially with the number of measured protein markers, manual identification becomes more and more challenging as there are too many combinations to consider. So the LabVantage analytics solution for flow cytometry can play a huge role to address some of these challenges. So the computational flow cytometry solution leverages AI and can help in combining novel algorithms for variable selection, clustering, cluster matching, feature selection. So AI actually offers methods and techniques for dimensionality reduction, cluster discovery on high dimensional data, and it helps in automated population identification and cell sorting using auto gating. There are even features for guided gating. Let me take you through a short demo.
Thank you.
Here is a short demo of the computational flow cytometry solution. Create a study to act as a folder to store the various FCS files associated with the certain project. Now click on File Upload and select the files to be uploaded into the study that we just created. Now you have all the files for the study loaded onto the system and you are all set to analyze these files. Explore the data using the data exploration feature of the solution. Try the cutting-edge AI algorithms for gating. Select the study and select the file. Then select the dimensionality reduction algorithm from a library of options. TSNE is a very popular choice and fill in all the associated parameters. Next, select the clustering algorithm. Again, there are a library of options. Specify the number of clusters of interest and then click on Run. You will get a notification once the execution is complete. The Results tab will show you the outcome of the run. On the left you will see the TSNE plot and so on the right you will see the outcome of the cluster analysis. It shows 4 clusters because we had specified 4 clusters as an input to the algorithm. This is an example of how our unsupervised learning algorithms are helping analysts with AI-based gating. The solution offers manual gating options along with a guided gating approach. Select the study and file that you want to gate and then select the options for X-axis and Y-axis. Perform manual gating with a lasso-based approach and create the desired sequence of case. On the left pane you can see the complete hierarchy of the gating strategy applied. At each level you can see the subset of data points selected. Once the gating for the first file is complete, save the gating strategy used as a template. This template will be the basis with which you can perform guided gating for other files in the same study. Choose the second file from the same study and click on the template to be applied. The solution automatically creates the 3 levels of gates for the second file using the template. The guided gating approach looks at the spatial coordinates of the gates as well as the relative positioning of the gates with respect to the cluster density. In this way we can order the remaining files of the same study using the template. The solution allows you to change the position or the size of the suggested gate. The template can learn from every manual intervention and keep auto-tailoring the gating strategy. The solution also has contour-based gating options. Event density is depicted with contour lines as the gradient. Closely packed contours indicate a high concentration of events. You can zoom in and the contour lines become more visible. You can choose to gate by clicking on any one of the contour lines. The solution will select all the points within that circle. To summarize, the flow cytometry solution caters to the various needs of researchers and analysts and potentially provides a 10x increase in the speed of analysis with significant cost reductions and a robust analysis approach that reduces bias from manual gating. Try the flow cytometry analysis solution today.
Wow. I'm truly impressed with the products that you've shown me so far. I'm sure the remaining products will be as good as these. One last request. Does LabVantage analytics of generative AI, or commonly known as Gen AI?
Yes, of course. I think you'll like it. Let me show you.
But before you start, wanted to lay out the context for you. As you know, I've been thinking about this for quite a while now, and talking to my colleagues out there, and they're all like going after these generative AI. So we don't want to fall behind. So as you know, drug R&D, manufacturing, commercial divisions, everyone generates tons of data, even at a single molecule or drug level, right? And most of the molecules in the R&D process that have failed, right? As you know, the probability of success even from IND to launch is 10%. And think about all the discovered molecules or patented molecules going through lead optimization, preclinical, etc. The probability of success is much, much lower. So all those learnings from those failed molecules and ongoing molecules that have not been launched, right? All that information coming through various divisions in the drug development continuum adds significant or contains valuable data and a lot of lessons learned that could be used in the iterative process. Okay, so question is, how do we combine and all this data and get accurate insights, whether combining the publicly available data and or internal data coming from all these different divisions? Is there a way to achieve something like that, or does LabVantage Analytics have something like that which can be achieved in minutes as opposed to several people working for months?
Yeah, so the solution that LabVantage Analytics has is absolutely the right fit based on this context that you just described. So let me highlight some of the key aspects. So as you know, ChatGPT will be trained only on publicly available data, and it tends to hallucinate. But ChatGPT does not have access to all the company's proprietary data. And as you mentioned, we have so much of data which is proprietary for us. So data on different toxicology reports, PK reports, data on all our failed experiments as well as successful experiments. And so ChatGPT, there's no way ChatGPT will have this information. So it will miss the critical information which is contextual for us while answering a specific question from our scientists. So tcgmcube's local large language model uses ChatGPT's trained large language model and further trains the model on the local and proprietary data sets for a company, which means it will further train itself on our data. So our tox reports, our PK reports, our data on our clinical trials, our failed experiments data, our regulatory reports, and any other journal or scientific papers that we may want it to get trained on. So references by ChatGPT, usually for every query, it's either at the journal or at institution level. But tcgmcube's local large language model has the option to give references for every query at a very specific article level and pinpoints to very specific publications. And this really helps set the context for our scientists. The value proposition is huge. we are looking at a 10x faster response on our searches. And we will also be looking at significantly improving the relevance, the contextualization of the information that's presented by the search. Let me take you through a short demo to illustrate.
The healthcare and life sciences ecosystem is highly complex, comprised of different stakeholders with different goals and objectives. Patient is at the center. The other stakeholders are providers who treat the patients and prescribes medication. The pharma or biotech stakeholder develops and sells those medications, and the payer, the health insurance companies, pay for it. In the pharma and biotech industry, vast amounts of data are generated daily across the entire drug development continuum, from discovery and lead optimization to preclinical and clinical development, commercial and manufacturing stages. Despite the staggering volume of data, much of it remains proprietary and unpublished, especially data from drugs in development and failed drugs. This information, although valuable, is often inaccessible to researchers and clinicians in general. Imagine if drug discovery scientists had a tool built on a large language model that could query this pool of information and extract relevant insights in no time. Access to this diverse data and intelligence pool could revolutionize their work. While open-source large language models like ChatGPT can provide access to the latest information, they are not trained on specialized and proprietary life sciences and medical data. Let me show you how. I am now in ChatGPT and I'm going to ask a question. Which mutations do children carry with juvenile myelomonocytic leukemia, JMML? As you can see, it provides 3 common mutations: NF1, PTPN11, CRAS, and NRAS. This may not be sufficient for a scientist trying to design their next experiment because they want more information. We are now going to open our own tcgmcube General AI Enterprise Search Engine, which is our large language model, and we are going to ask the same question. In fact, we did get all those answers that ChatGPT gave, but it has more, such as SETBP1 and JAK3. The query also returns the references where this information came from, because the local LLM model was trained on a subset of information. The references being a publication with PMID ending with 7648 provides the basic information, and another publication with PMID ending with 2011, which shows a whole exome sequencing that was done which identified these secondary mutations SETBP1 and JAK3. While the ChatGPT query did not provide this information, but it showed up in our local large language model. We have trained our local large language model on highly specialized scientific and medical information that we have been collecting over the last 2 decades. Data on protein structures, chemical structures, drug labels, clinical trials, patents, genomics, clinical data, other publicly available data. Over 3.3 million biomedical entities, 440 million relations, and 40 million documents have been mined and curated. Not only that, but global knowledge graphs have also been created using AI by creating the ontologies, contextualization, applying the semantic modeling, and fairyfication. And that's why when tcgmcube's local LLM was used, it applied the knowledge graph and the semantic search and returned all the necessary information as the query result versus ChatGPT. Let's try another query and see what answers are generated by ChatGPT versus our local large language model. The major genes associated with Alzheimer's disease include these 6, as well as some information related to it. While this response is extensive, it may not be sufficient. I'm going to run the same question in our local large language model. You can see it has returned those listed in ChatGPT, but has also returned more along with the references. Let's go to the references, the genetics of Alzheimer's, which was published in 2012. It has the basic information. Also, let's go to this reference on rare coding variants associated with the Alzheimer's, published in 2017, where it talks about the rare coding variants PLCG2, ABI3, and TREM2. So, this is another example how not just the main genes, but also the rare variants are captured as responses in our local large language model. And this is because of the semantic search that is applied using the ontologies. In summary, it's fair to say that open source LLMs not just hallucinate, that is giving false positives, but also misses reporting critical information, false negatives. Missing such critical information, which we call known unknowns, can be as detrimental as hallucination. Our local LLM has been trained on our owned and specialized data while open source trained on publicly available data. Now, your proprietary data such as toxicology reports, PK reports, clinical trial protocols, regulatory reports, and other proprietary documents for successful, ongoing and failed molecules can now be used to train the local large language model to provide faster and higher accuracy in responses and bring in more productivity to your enterprise and industry search with high velocity. Our application pinpoints the specific articles taken as references, whereas ChatGPT provides references at an institution level.
Okay, I'm fully convinced that LabVantage analytics products are superior in terms of its functionality, ease of use, and AI capabilities. But for me to justify the investment, I'd like to know the value propositions in terms of time and cost savings. Have you done that part of the exercise, Panchali? Would like to hear from you.
Yeah, sure. Let me talk about the value proposition. So the integrated solution can offer huge benefits to our R&D labs. My calculations show that we can reduce our analysis and validation of efforts by over 50%. And of course, this will lead to huge cost savings for our labs through higher productivity and data-driven recommendations for our analysis.
So final question, I'm good with this. You know which customers are using LabVantage analytics products? Do you have a sense of that?
Oh, this is being used by some of the top 50 pharma biotech companies, and it has a global footprint. So, we have companies, global companies using this, so very widely adopted.
Okay, I'm good to take this to my boss, the Senior Vice President of Research at Cutting Edge Pharma, and secure the funding for LabVantage analytics. You now have a tougher job to do, Panchali, to convince the current scientists in your lab to switch. But I know you'll make that happen. All the very best.
No problem at all, Krish. The nature of the solution makes it very easy for user adoption. It is super interactive and offers an excellent user experience. So it will actually be quite easy for me to talk to our colleagues and get them to accept the new solution.
Okay, so for the audience, you know that we flipped our roles. Now we are back to TCG Digital and LabVantage. So this ends the role play. Hope this helped to make this webinar interesting and useful for you. We are now ready to answer any questions you may have. Thanks again for the opportunity and your dedicated time.
Okay, so I'll be moderating the question and answers. That was a very engaging session, Krish and Panchali, especially with the product demos. Let me just stop sharing my screen, and we have a very engaged audience as well. They've been sending in questions. So here is the first one. What are some of the standard reports that are provided in the assay analysis solutions?
So let me take that. So the assay analysis solution, there's a wide assortment of reports that can be provided, right? So we have this whole reporting solutions, and it depends on the assays. For example, some of the reports for PK assays will include things like intra-run precision and accuracy analysis, or inter-run precision and accuracy analysis, integrity of dilution, dilution linearity, freeze-thaw stability, benchtop stability, frozen storage stability. Now, if you're talking about ADA assays, that will include, reports such as overview of system suitability results for analytical runs, reported sample results in the titer assay, sample subject to reanalysis due to analytical anomalies in the titer assay, similar results for screening assays, and so on. So there are quite a few reports depending on the assay that we are speaking about.
That's great. Thank you for your response, Panchali. And here is the second question. She says, I have a question regarding the application of AI and LLM in formulation science. I do understand the application and analysis. However, how can we minimize or rather optimize our time to develop formulations?
So I would say that this is a great question, and we'll be more than happy to look at the specific use cases that you are mentioning to see what information that is needed for your particular use case, and we can develop a strategy to help you with the local large language model development. It's all about the information that's needed for you to build the next, in this case, the next set of formulations, right? And for that, you may have many different documents embedded or archived, which you have to kind of do manually or go through it through some other means, but not get those answers accurately within a few minutes. And this is where the model comes in. We look at your documents, we train the model. We still use all the information out there in the public domain, but as you saw in the video, that we do gate all the questions within a knowledge graph, as opposed to letting it go free flow everywhere, because scientific information or medical information are not easy to annotate and curate, but we have already done that. So once we have that information set of documents, we'll bring that local large language model and train against your data, and we should be able to provide you answers. We'll be more than happy to catch up with you offline and discuss this in more details.
Thank you, Krish. Thanks for the response. The next question is more around how are assay analysis solutions helping in drug discovery overall?
Okay, let me answer that. So this is all about speed and reliability. So one integrated platform for any assay method validation, sample testing, and reporting. So the integrated solution can save companies millions of dollars of effort and accelerate the race for new drug discovery. So complex assays further leverage AI to gain deeper insight. We saw some of that in flow cytometry, because traditional approaches for analysis will not adequately cater to the high dimensional panels. So all of these significantly expedites the need for, expedites the analysis effort, and that plays a strong role in expediting a new drug discovery.
Okay, our next question is on flow cytometry, the demo that you showed. What are some of the algorithms that are being used in flow cytometry?
So the flow cytometry, it actually has multiple algorithms. Let me just speak about a few. So if you're talking about dimensionality reduction, it's using, currently the repository has algorithms such as TSNE, UMAP, principal component analysis. And if you're talking about clustering, the clustering algorithms in the library, so I'll mention some of those. So SOM or self-organized maps, Gaussian mixture models, k-means, Leiden community detection, a phenograph. There are quite a few.
Okay, great. Thanks, Panchali. We have time for one last question, even though there are, audience who are sending in their questions in email as well. But please be rest assured that we will get back to you over an email. So we'll just take one last question for this session for Krish and Panchali. Do you also provide a solution for qPCR and RT-qPCR?
Yes, of course. We have solutions for both qPCR and RT-qPCR. Let me talk about some of the features of these solutions. So you can fit the standard curve with log of quantity as X and your CQ value as the Y. So you can use the computations to use the curve to generate the log of copies per reaction for standards, for unknowns. And you have the options for normalization and different options for outlier analysis. You can do unit conversions. You can apply all your specifications as with the other assay analysis solutions. And you'll get your outputs for your curve coefficients, summary statistics. And of course, the solution has bidirectional integration with LIMS. So you read data seamlessly from LIMS and you write back the outcome to the LIMS system for analyst action.
Great. Thank you so much, Panchali. Krish, do you want to add anything to that or an overall concluding, message from you?
Absolutely. No, thank you. First of all, thank you for the opportunity. I know that we have participants from all over the world listening here. And this is not the end of it. We have your questions, and feel free to reach out to us anytime. We should be able to provide you answers or set up meetings to discuss. But when we talk about the future, right? Information is so critical, and millions of documents across the globe, even for a given clinical indication, is generated on a monthly basis or even daily basis. And it's almost humanly impossible to get all that information in your head or a group of people's head to derive something meaningful. And that's why, whether you're doing target identification in the early discovery or lead identification, lead optimization, or even if you're doing translational research, you're trying to predict. This is where we have to really rely on all that information, not just public, but more importantly, your experiments that you have developed over time, whether for your molecules that are ongoing or backup or follow-up molecules or failed molecules. That lesson learned is very important to bring it back to discovery or early stages of drug development continuum. And for that, you cannot wait months. You have to do it in days or even minutes or hours. You need that information so you can set up the experiment and move forward. This is where I think we can add significant value. And we have trained a lot of these local large language models with companies, and we have a lot of data science competencies, as I was mentioning, including scientific and PhD data who have been helping develop these local large language models. And finally, as Panchali mentioned, that as you're using the LabVantage LIMS, which is, you have to use LIMS when you're in a lab environment, it's always good to know that there's a seamless way to connect the analytics and not take the data out and use any third-party tools. That adds to a lot of errors, a lot of mistakes. You have QA issues, Part 11 issues. So all these could be bypassed, and it will not only save time but also add significant value. So once again, thank you for the opportunity. This is a great session, and we should do this more. Thank you.
Thank you.
Thank you. Alan, do you want to kind of just wrap it up?
Yes.
At the end?
So thank you all again for participating. And as Dr. Ghosh said, if you have any other questions, please contact us or your local LabVantage representative about LabVantage analytics or anything that we offer here at LabVantage. So thank you again for making the time, and we'll be speaking with you soon.
Thank you all.